Good morning, everyone. Thanks for being here. It's very chilly outside, so I know it's tough to get up at 8:30 and be here, but it's my great pleasure to introduce Dr. Christos Davatzikos from University of Pennsylvania. Dr. Davatzikos is the Wallace C. Miller Senior Professor of Radiology and the Director of the Center for Biomedical Image Computing and Analytics. He holds a secondary appointment in electrical and systems engineering at Penn, as well as bioengineering and applied mathematics graduate groups. He obtained his undergraduate degree from the National Technical University of Athens, Greece, and his PhD degree from Johns Hopkins in 1994 on Fulbright scholarship. He then joined the faculty in radiology and later in computer science, where he founded and directed the neuroimaging laboratory. In 2002, he moved to Penn, where he founded and directed the section of biomedical image analysis. Dr. Davatzikos' interests are in medical image analysis, and he oversees a diverse research program ranging from basic problems in image pattern analysis and machine learning to a variety of clinical studies in neuroimaging disorders, brain cancers, and other brain development disorders. Dr. Davatzikos has served on a variety of scientific journal editorial boards and grant review committees. He's also the IEEE Fellow and Fellow of the American Institute for Medical and Biological Engineering. Before I hand over the mic to him, we do have a little gift for him as a token of appreciation for coming all the way. We really appreciate it.
Thank you so much.
Thanks for being here. With that, I'll hand over the mic to Christos.
Great. Thanks so much, Pallavi. It's a great pleasure to be here, and thanks for getting up so early in the cold. It's really chilly outside. Let me see if I can find... I'll talk a little bit about, I know it's a very diverse group and center. I will talk a little bit about our work on machine learning and neuroimaging. I spent the first part of my career in doing medical image analysis back in the 90s and early 2000s, developing algorithms to extract features, to register images, and then I realized that machine learning is it. We have enough ways to extract features, so we have to tie it together into higher level imaging signatures, if you want, of diagnostic and predictive value, and that's what I've been doing for 15 years now. Machine learning has, I used to work hard to convince people that it's a good thing. Now, and everybody wants to do machine learning, so it's a good time. It's an easier time. But most of the time, or much of the time, you hear machine learning being used for things that we can actually do as humans, such as segmentation, but helps automate things. So there's a whole bunch of people working in this area, but I'm more interested in these tools helping us see what we don't see in the images rather than see what we see in the images and do it automatically. And this is what I will be talking about, particularly in two fields, clinical neuroscience and neuro-oncology and brain gliomas. So this is an example from some of our work in schizophrenia. You see here an MRI scan of a person with schizophrenia and a person of the same age who is normal control. There is really, this MRI is called completely normal. Any neuroradiologist would call this MRI normal. However, this person has a devastating disorder. Something must be abnormal there that we don't see. If you start measuring brain volumes of the cingulate gyrus and other parts of the brain, you start seeing slight shifts here. The patients have slightly smaller volumes in this and that and the other region, but none of them is really the magic bullet that will trigger this scan to be called abnormal. However, the combination of them forms a pattern that makes it, as I'll show you in a second, quite abnormal. Same thing for people who have mild cognitive impairment. This is an MRI of a lady who's the same age as this lady here, but she has a cognitive impairment, and statistically she's very likely to progress to Alzheimer's disease. Something is different in this scan that we don't see in this scan. But we don't see. And again, if you start measuring volumes a little bit, you will see the hippocampus, the entorhinal cortex, and a bunch of other areas are smaller in people with Alzheimer's disease, but in people with mild cognitive impairment, they're just slightly smaller, and none of them really gives us a sufficiently sensitive and specific diagnostic tool. And in neuro-oncology, some of our current protocols, clinical protocols, have become very complex. So with every glioma patient goes through an extensive protocol of standard MRI plus some perfusion scans, diffusion tensor images. You have all the derivatives from that. The dynamics of perfusion can be captured using temporal models and so forth. So there's a lot of information here. And up to now, most of the time, people clinically would just look at the tumor, will measure how big it is or whether it enhances or not, and so forth. But there's so much more information here that allows us to tell if someone has EGFR or IDH1 mutations or MGMT promoter methylation from these scans. There are hidden patterns in the data that allow us to tell with a fair amount of accuracy. Is this patient going to respond to standard treatment, and what is the expected survival? And if not, should we put them in different treatments up front rather than waiting for the treatment to fail and so forth? So all these are patterns that we don't see, but they can be appreciated better with machine learning. But another aspect of machine learning that I'm particularly interested in is that of dimensionality reduction, distilling complex multivariate patterns, imaging patterns down to a small number of informative biomarkers. Imaging data has become very complex. And it's often difficult to make sense out of it. We want to distill it down to a small number, and there's an effort that I will come back to this slide in a second, but I just want to tell you a little bit of the motivation. We want to use machine learning to distill this complex data. We have put together a consortium of about 20,000 brain MRI scans from multiple studies, and we're trying to understand, to come up with better understanding of patterns of brain atrophy in mild cognitive impairment and Alzheimer's disease. These are a few dimensions here. You might have this pattern or this pattern or that pattern. It turns out that they have different outcomes. You might have a different pattern of amyloid deposition, and that's predictive, a different pattern of functional connectivity. All these are maybe 20 or 30 dimensions that now allow us to distill this very complex multimodal data down to a small panel that can be potentially clinically useful and informed by big data analytics. So machine learning, again, a lot of people here use it routinely. We're sampling a number of different brain regions and a number of different signals and features that we extract from them to come up with something that, if it's positive, an index that if it's positive, you're expressing this pattern. If you're negative, you're not expressing this pattern. So we've been calling some of these indices SPARE scores for spatial patterns for recognition of something, SPARE-ED, SPARE schizophrenia, and other patterns. This is the typical pattern we have found in Alzheimer's disease patients that involves the hippocampus, temporal lobe regions, and a bunch of other regions that you don't see here, the posterior parietal lobe, perihippocampal white matter. Schizophrenia has a very pronounced frontotemporal pattern of brain atrophy. Again, none of these regions is sufficiently atrophied for us to be able to see it visually. Do you hear me without the microphone? Yeah, okay, so that's better. So none of these regions is sufficiently atrophied. They won't be able to hear you. Oh, they won't. Okay, all right. But the combination of them makes a pattern that is fairly distinctive. So, and then it is distinctive, as you see here, the people who have Alzheimer's disease have positive scores, the people who are controls have negative scores. So this almost entirely perfectly separates the two distributions. So we have an index now, the SPARE-ED score, that is very, very sensitive and specific, and gives an ROC of this sort. And it is one of the dimensions so in our coordinate system, if you want. Now, of course, you don't want to have an MRI for someone who has Alzheimer's disease in order to confirm that. You're interested in going back to prodromal stages, to preclinical stages. So this is, we evaluated in a study a few years back this SPARE-ED index in people with mild cognitive impairment. And we asked the question, do people who have good scores at baseline have better outcome than people who have poor scores at baseline? And it turns out that they give us pretty good survival curves. So we can predict, based on the baseline scans, whether or not someone is likely to progress to Alzheimer's disease from MCI. And if you add a couple of scores, a standard cognitive score and the ApoE4 genotype, which the ApoE4 is the biggest genetic risk factor in Alzheimer's disease, you have a very good separation. So now, at baseline, we can predict whether or not you have, if you present to the clinic with mild cognitive impairment, you can predict fairly well if the patient is going to have a good outcome or poor outcome. And of course, that brings up the question, what if you go earlier to normal brain aging, years before people become demented? It turns out we had a paper in Brain in 2009 that together with a Baltimore study of aging, it's a longitudinal study of aging, a prospective study, we found out that the change, the longitudinal change, this is the rate of change of the PRED score, the rate of change at which we accumulate this specific pattern of brain atrophy that we have found in Alzheimer's disease. The rate of change separates people who finally progress to MCI, to mild cognitive impairment, from the ones who don't. You see that these people are almost all above the line. These people are most of them below the line. Unfortunately, everybody has positive score. Things don't get any better as we get older, for any of us, it seems. But for some people, the rate of change is slow enough to be putting us on the safer side, if you want. So this is a prospective study of, I would call, super normal, super healthy people. And they were been followed for many years. So you're looking at detecting these patterns with these machine learning tools potentially 20 years before someone gets demented. That's a pretty good time to detect things. That's the window of opportunity for treatment. There are no treatments right now, but at least you can control comorbidities such as hypertension, diabetes, and other things, lifestyle changes. If you're showing a pattern of brain atrophy that seems to be progressing towards MCI and later to AD, you can potentially be more aggressive on the comorbidity side. But it's interesting that if you look at these people 20 years before they get demented, you'll see absolutely nothing, a perfectly normal scan. But the machine learning tools pick up these subtle patterns very early. Same thing with schizophrenia. As I said earlier, normal MRI, most people, would attest to the fact that the brain MRI of a schizophrenic patient is normal, but unfortunately there is a subtle but pretty distinctive pattern of brain atrophy that involves certain prefrontal regions, certain temporal regions, perisylvian regions, and this is a study, another consortium that we've put together from people in Europe, in China, and in the US. This is about 700 or 800 people, and we found very good compatibility across ethnic backgrounds, scanners, and continents, if you want, of this imaging signature that allows us to obtain an ROC curve that is around 0.83 or so. We're trying to get this a little better using deep learning tools and things like that now. But it turns out that we have a neuroanatomical signature of schizophrenia, and the question is how predictive that is in first episode people who have variable outcomes. I was going to talk. There are some challenges that we face. So this is one of the challenges for those who are statistically interested. Machine learning tools are usually called, thought of as black boxes. They don't allow us to interpret easily what information contributes to the learner. This is some work one of my students, Bill Wozgankar, who's now at UCLA, did a few years ago in his thesis, where we actually came up with mathematical analytical forms for estimating statistical significance of different features that contribute to linear support vector machines. This is a fairly simple classifier, but in that case, we can actually not only come up with the right answer, but also highlight parts of the brain and features that are statistically significant in terms of contributing to the classifier's output. So I would like to point out that sometimes these machine learning tools require a very large very large sample size, and it might not be easy to learn a global pattern of, say, schizophrenia or Alzheimer's disease, but you can apply these things locally as well and find regional patterns and trying to stitch them together. And actually, we do these kinds of things. How many of you use statistical parametric mapping, SPM, and voxel-based morphometry? And some people, not many, okay, all right, some people do. So the first thing one does is to apply a local filter into the data because voxel-wise data is too noisy, there are registration errors, we want to Gaussianize it, and so forth. And typically you apply a local Gaussian filter of maybe 4 millimeters, or maybe 8 millimeters, or maybe 15 millimeters, who knows? Everybody has their favorite filter, right? The question is, why should it be fixed throughout the whole brain? Why should it be Gaussian? Who is to say that the Gaussian filter is the best? And what should the size be? If you actually take a study of schizophrenia patients versus controls and you apply a standard voxel-based morphometry with 3 millimeter, 6, 10, 15 millimeter filter sizes, you'll find different results. These are the same people. The same exact analysis, it's just we smooth the data by different filters up front. Well, you get, I mean, not dramatically different results, but the conclusions change quite a bit as you do that. So who is to decide what the optimal filter is? Machine learning actually can help us do that. Maybe the part of the cortex needs to be filtered with a large filter of this sort, and maybe part of the insula should be filtered with such a filter, and maybe the hippocampus should be filtered with such a filter to best tease out the signal. This is basically exactly what machine learning does. Machine learning is at least a linear classifier would be a linear weighting of different features. And that's a filter that has been optimized to maximize the difference between two groups that you're comparing. So if you want to compare schizophrenia patients and controls, if you apply a local linear support vector machine or something like that, the weights of the different voxels would be a local filter that is optimal. And maybe the best filter here is a big filter that looks like this, and the best filter here is a filter that looks like that. And that teases out the signal much better. And that's what we do. This is another former student of mine who is now in New York, Erdem Varol, who thought of using local machine learning local windows. He throws thousands of windows around the image. He has a local classifier. He somehow stitches them together and combines the weight. And then I'll skip the math here. We come up with a map of statistically significant regions of difference between patients and controls, progressors and non-progressors, any two-group analysis or regression. But it regionally uses the optimal filtering of the data using regional machine learning tools. And that performs much better than any other alternative we tried, including the Searchlight, the standard statistical parametric mapping, the GLM, and so forth. So you don't have to have a huge N to apply these tools. You can apply it on a regional basis and then stitch the results together. Another challenge that I've been particularly interested in the past 4 to 5 years, and we've done some work on that. This is my former student now, Jan Dong, who developed a method for heterogeneity analysis. So a machine learning tool assumes that there is a pattern that differentiates Alzheimer's from controls, progressors from non-progressors, and so forth. But there's not a single pattern. What if you have heterogeneity in disease? What if you have multiple subtypes of disease? And there's not a single pattern that separates, say, patients from controls. There might be multiple. So this heterogeneity is very, very important in a number of contexts. And we've developed some semi-supervised learning tools that allow us to estimate not a single pattern, but multiple patterns that take, say, measurements from a number of from a control population and map them to, say, a patient population. So the idea in these semi-supervised learning methods is that you know that these people are different from these people. You're sitting now in a high-dimensional feature space here. And these are patients and these are controls. But you don't know which of these people have better outcome and who have worse outcome. You don't know who have potentially be affected by one neurodegenerative process compared to another one. But you don't know, you do know that they differ from these people who are cognitively stable. So we have some math behind it, and I will skip that, but basically we'll come up with multiple subtypes of disease. And this is an application. We had a paper in Brain last year, or the year before, where we applied it to data from mild cognitive impairment and Alzheimer's disease. We put together all the MCIs and ADs in one bucket and then compared them with normal controls. And we asked the question, how many patterns of neuroanatomical difference do you see between these two populations? This is from ADNI-1 and ADNI-2, which is a fairly selective study. So you expect a fairly homogeneous group, but nonetheless, we found 4 subtypes of people with MCI here, which was, even I was surprised. I know the heterogeneity is a big issue, but there was a 13% of people who were declined, but they didn't have any neuroanatomical difference from controls. There were 44% of the people who were either, who had the typical pattern of brain atrophy in hippocampus and medial temporal lobe that is expected in Alzheimer's disease. So this had the pattern that we expected to see. It was only 44%. There was a 15% of people who had a very focal atrophy, very normal brain elsewhere, but very focal atrophy in the regions that we know that are affected by Alzheimer's disease. Again, the hypothesis here might be that these are at early stages of progressive disease. And there was a 28% of people who were the most interesting, in my opinion, who did not have this pattern. So these people were either demented or they had mild cognitive impairment, but they didn't have the typical Alzheimer's type of brain atrophy. They had a more diffuse pattern of atrophy. And if you look at survival curves, you see that the people who had the most typical AD pattern of atrophy, they would decline fastest. The people who had this focal atrophy initially were normal, not normal. Initially, they had the very slow rate of progression, but then they took off and they almost reached these guys. So these people perhaps are people who are at early stages of progressive and aggressive disease. And these people who had this unusual pattern of atrophy, half of them leveled off for a long time. They did not progress to Alzheimer's disease. So that might be indicative that they have something else, or they have mixed pathologies. But certainly these are people who are different from these people. So all of these now, right now, clinically, are lumped into one bucket of AD or MCI, but they have different characteristics, and potentially down the road we should be looking at them separately. So we want to put all this together. I said before, we've collected data from now 20,000 people. I mean, this is already collected data by various collaborators. Some of them is public domain data, like ADNI and UK Biobank. And we put it together. We have developed statistical harmonization methods to be able to create comparable measurements. And we want to come up with what we call I-staging. It stands for something imaging-based coordinate system for something for aging and neurodegenerative diseases. But basically, we want to have a few dimensions. These are the subtypes of atrophy we see in MCI and AD. So maybe these are 3 different dimensions. A few other dimensions relate to similar patterns of amyloid deposition that we found. People deposit amyloid in slightly different patterns, and that might be important. Patterns of brain connectivity and so forth. And we want to come up with maybe 20 or 30 dimensions to be able to take anyone's brain MRI scan and put it in this coordinate system and ask the question, where do you stand with respect to this large and diverse population? And based on what we've seen in terms of cognitive scores, in terms of clinical progression, what are your odds of, given that you have this brain MRI scan, what are your odds of progressing to decline cognitively or to become demented and so forth? And as we put more studies there, we have ancillary studies of diabetes, so we want to put of hypertension. So we want to put all of these studies in this coordinate system and understand what, say, aggressive treatment of hypertension do to your brain MRI measurements. How does it modify your trajectories in this standardized reference system? And I think that's clinically appealing because if a clinician has to deal with 100 million voxel short time series points in a time series, it's very difficult. If you have to make arbitrary decisions of ROIs, why this ROI and not that ROI, at least now we have a way to condense this data down in some way that has been informed by analytic study of this scale. Now, what you can do in this coordinate system is start visualizing things, and some of our clinical collaborators love these plots here, where you can, I cannot plot 20 or 30 dimensions, but I can plot 2 or 3. Here I'm plotting the amount of this white matter hyperintensities, which are small vessel ischemic disease, age, and the color map is the brain age. So we put the data together. We're predicting someone's brain age. Of course, the idea is not to use MRI to predict someone's age. You have their ID card to see that their birth certificate. But the idea is to find out the residuals. Does your brain look older than expected or younger than expected? And to understand what these residuals mean. So what you see here is that, say for a 60-year-old who has a low amount of this ischemic disease, of the small vessel disease, they look like a 60-year-old. But if they start accumulating some of these lesions, which are known to be related to hypertension, to obesity, to diabetes, and a number of other factors that increase the presence of these lesions, which are, by the way, a lot of people will call them normal for the age. If you ask a neuroradiologist to read a 60-year-old's MRI scan, they expect to find some of these ischemic small vessel disease changes. Then if you accumulate that many, you start to look like a 65-year-old. And if you accumulate a lot more, you start to look like a 70-year-old. So you start to see how this accelerates your brain aging. And these isocontours, these are isocontours of the brain age. There's an underlying spline model that has been fit to them. They become steeper and steeper as you become younger. So the younger you are, the more important, the more detrimental the presence of these ischemic small vessel disease lesions is. Whereas if you're older, it's not that they're not important, but at this stage, at the stage of at the age of 80 or 90, a lot of other neurodegenerative processes have kicked in, including of course amyloid plaques and tangles and a number of other things, that these ischemic lesions might be a little less important in terms of pushing your brain age. Another plot here is that, again, a different view of this system where I plot the y-axis is the spare AD score, is the AD-like atrophy. This is the brain age, and in the color map, the color scale is the trail-making test. It's a cognitive test that is primarily teases out executive function. And as you see here, the isocontours start to tell us that as you get older, you get worse at this test, and as you get, and for old people, if you start accumulating AD-like atrophy, then it pushes your performance quickly up. So again, at younger ages, it is your brain age score that is important in terms of cognition, but at older ages, and if you start accumulating patterns of atrophy that are typical in AD, then those become more important in terms of pushing your cognition down. Again, this is a way to start exploring the data in a way that teases out some of these relationships between brain age, AD-like atrophy, and executive function. And finally, some of the applications in neuro-oncology. This is a different area we've been working on, but I think it's also very exciting. And there's a lot of work here, and Pallavi and her group and others have very extensive work in this area. I've been working with brain tumors since 1997, and I was a believer in these types of methods, obviously, for 20 years. But even I have been surprised in terms of how much information is hidden behind these images that we don't see. And as I was saying earlier, current protocols are quite complex. Our glioma patients typically get the T1 GAD, T1, T2 FLAIR images. We get the perfusion series, which of course is a time series that you can extract things like RCBV, the relative blood volume and blood flow, but you can extract other things using dynamic models. You can extract information about how these curves change over time with the administration of the bolus, and then and that relates to leakiness of the vessels, to vascularization, and so forth, to perfusion, to shunting, and other things. You have the diffusion signals, the diffusion tensor imaging that, of course, gives you various derivatives. The radial diffusivity relates to cell density. You have fractional isotropy that relates to the microstructure of the tissue and so forth. You have spectroscopy. You have models that you can fit to the data. So there's a lot of information. And right now, or up to now, clinically, people measure how big the tumor is and whether it enhances or not, which throws out 99% of this information, basically. It turns out that I'll show you that we can tell a lot more by these images. For example, we have shown in a study of 90 people, initially it was 65 or so, and then we had a subsequent follow-up study. We've shown that you can predict where the tumor is infiltrating and where it's likely to recur. This is the tumor here, and this is the peritumoral edema, which is this is a very small edematous region. Sometimes it's a huge area, this is the infiltration front. And like we like Pallavi's group and others have found that there is a lot of information in that infiltrating front that is perhaps more useful than the tumor itself. So, but in any case, we're looking at this area, and we put together all the signals that we have. We've looked at follow-up scans of people who recurred, and we found that you can go back to the baseline scans pre-surgery. These are images pre-surgery, and you can create maps like this that predict where the tumor is likely to be infiltrating and where it is likely to recur. And if you follow them after resection and for months later when they recur, in many cases the recurrence occurred where we had predicted it. Again, this is a statistical phenomenon. This is a statistical map. It doesn't tell us exactly where the tumor will come back, but it tells us that this tumor has, based on our expectations, is approximately 10 to 15 times more likely to recur than this tissue. And we've done this extensively in different ways. So this is significant enough for people for our radiation oncologists at Penn to start a clinical trial, which is ongoing right now, and it delivers 75 gray to these red hotspots or red areas instead of 60 gray, which is what is the usual treatment is for the peritumoral ring of some millimeters. And we want to see if that is going to delay recurrence. We also have a second trial, and Andy Sloan was here. He just left, but we have a collaboration where he's trying to put together another clinical trial at the national level where they will resect extensively peritumoral tissue that is likely to recur. And again, this might alter clinical outcome. This is not a slide for computationally savvy people, but a lot of times when I talk to our clinical collaborators, I use this to emphasize that these probability maps of infiltration are not influenced by any single magic bullet. There's no single signal that tells us that the tumor is infiltrating. But there are subtle differences in a number of different signals. This is a T1 GAD. Red is the people the voxels that presented recurrence, and blue are the voxels that did not present recurrence. And there are slight differences in all of them. Slightly elevated T1 GAD, slightly lower FLAIR signal. Again, this should be consistent with slightly elevated microvascularization and leakiness of vessels, slightly maybe denser tissue, less water content. T2 is smaller. The trace also is consistent with more dense tissue in the areas that recur. These are the principal components in the RCBV of the perfusion signal. The second principal component has some information there. So there's a little bit of information hiding in all of these. None of them is a magic bullet, but if you put them together, you get an index that is quite, using machine learning, you get an index that is quite distinctive. And this is not different from designing a ligand. Say this is a PET ligand that binds to a amyloid peptide, for example, and it has a certain form. There's nothing specific about an oxygen atom or a nitrogen atom or a cyclic route, but if you put them together in a form that is so specific, it binds to amyloid beta. Or it binds to certain mutant, certain receptors in the tumor and so forth. So it is all in the pattern of many, many things that contribute to something that becomes unique by virtue of accumulating a lot of these weak signals into a strong signal. We have ways, and that's very exciting. We can predict who is going to survive longer or not. Glioblastoma is a terrible disease. The median survival has been about 14 months. It has been slightly elevated now with what they call the tumor treating fields, but it's a very difficult treatment. You have to wear this hat all day long. So that prolongs your life for a couple of months. But some people live for 3 years or perhaps longer. Some people survive only for 3 months. And you'd like to know that up front for many reasons. Number one, you could factor that into a clinical trial statistical analysis to see if a treatment works. If you have a phase 1 trial with 20 patients, an early phase trial with 20 patients, you don't have enough statistical power to detect treatment effects, and all of these trials fail. However, if you have people who have such a diverse survival from 3 months to more than 3 years, then it's very difficult to detect a treatment effect unless it's very strong. But if you have predictions of survival as a baseline measure to compare against, you can normalize your measurements and have much higher statistical power. Also, if you have predictions of survival up front, and you know that the patient is very unlikely to survive long, then you might not want to put them in the standard treatment, but you might want to put them directly into an alternative trial that might benefit this person more. But you don't want to put these people to this trial because they are the ones who will benefit most from the current treatment. So we have, again, used machine learning, and we use a panel of maybe 1,500 or so features. And again, Pallavi's groups and Satish's group as well, they use similar features. We extract numerous features from different partitions of the tumor, from the peritumoral edema. We have deformable registration that map things to standardized atlas space. So we looked at the spatial distribution of the tumor. That is important. And we put together all these features, and we have come up with a predictor of survival, which is pretty good. This is prospective or cross-validated predictions of survival. And you see that at baseline, our prediction by putting together different signals can tell us whether or not someone has poor survival or longer survival. This is something we published a few years ago in neuro-oncology. What is very interesting is also that you can use unsupervised clustering methods, and this is a radiomic map of our glioblastomas. And we found out that there are 3 different subtypes of glioblastoma. People in GBM know that there are 3 or 4 molecular subtypes, but we also now know that there are different imaging subtypes that are very different. Each row is a feature, each column is a patient. And you can see these people here are very different from these people and from these people. The question is, do they have different survival? Well, this is survival separated by IDH1 mutation. IDH1 is a very important mutation in glioblastoma. It's clinically right now used for prediction. People who have the mutation have better prognosis. People who don't have the mutation have poor prognosis. And these are the 2 buckets basically that they use clinically. But what you see here is if people do not have the mutation, in other words, they have the poor prognosis according to the WHO classifications right now, but if they belong to this subtype, the imaging subtype, they have much better survival. These are the green circles here. The open circles were still alive at the time. So there's a big difference in terms of having this imaging subtype compared to having this imaging subtype. That now points to the fact that imaging can play an important role in these WHO classifications beyond what the tissue can tell you, because you don't want to tell a person that doesn't have that doesn't have IDH1 mutation that they have poor prognosis if they belong to this subtype. These are visually the subtypes. This is the better prognosis subtype. This is the worst prognosis subtype. And this is not something we expected. This sort of looks like a good tumor, although it has a big edema. It's a more circumscribed tumor. It doesn't seem to be infiltrating too much. But these are the tumors that we thought had the worst prognosis before. These are the people who had actually intermediate prognosis. This is this group, the blue group. The red group is this group, which doesn't necessarily look bad, but it is bad. And the reason why they have very poor prognosis is that it turns out that the tumor, sometimes being circumscribed and having a small enhancement, it seems to have very solidly infiltrated the peritumoral tissue, the brain. So it is the peritumoral tissue that gives away that this tumor is very aggressive. It has very dense and vascularized peritumoral edema, and therefore it's likely to have the worst prognosis. These are also the people who might benefit from extensive resection, because if we know that these tumors are so heavily infiltrating, we will have more extensive resection. So again, these are ways in which imaging can influence treatment right here and right now. What was even more surprising is that we can tell whether or not someone has a mutation. We have looked at 3 different molecular targets, and we're looking at many right now. This is EGFR V3 mutation. It is a mutation that is very important in glioblastoma because this has potential treatments, including immunotherapy. And of course, what happens right now is that they take the tumor out, they take one sample, and they analyze it in the lab, and they tell you whether or not you have the mutation. The question is there an imaging signature that can tell us whether or not you have the mutation in vivo? It turns out that there is one, and we achieved accuracy of approximately 90%, which is as good as the accuracy of the genomic test itself, essentially. So imaging carries all the information we need to know, perhaps more, because it captures the heterogeneity. So there's a number of different imaging signals that have been put together. You know, some aspects of the perfusion, the diffusion. I'm not going to comment too much on these, but there was one aspect that was extremely surprising. Came out of this and they came out of our ability to map all these tumors to a standardized atlas space and evaluate the spatial distribution of the tumors. It turns out that the people who had the mutation their tumors are in the posterior parietal lobe and some other areas, some of them occipital, whereas the people who didn't have the mutation had the more standard superior temporal gyrus pattern. And other areas, of course. This is a probability map of having the tumor at a given location in a standardized space. So you see a marked difference between these two. Now that we've seen the statistical atlas, we know that if the tumor is here, it's almost certainly negative for EGFR V3 mutation. If the tumor is here, it's much more likely to be mutant than not. So just the location, actually, now of the tumor becomes a very important feature. This is something that we never appreciated before. People thought that the glioblastoma was all over the place, and nobody would have predicted that the location would actually be associated with the presence of the mutation. So this is, again, the power of machine learning and big data analytics, that it teases out relationships from the data that are not obvious to our eyes. And a lot of this has been implemented in CAPTK, our cancer imaging phenomics toolkit, which is something that is open source, is free to download, it's developing right now, it's evolving right now. We're trying to incorporate a lot of these tools for cancer imaging analytics into this tool. We want to try Pallavi's collage features. I have to check with the guys if they've tested that, certainly, the more features, the better off typically you are. So that's all I had to say. In conclusion, machine learning teases out imaging patterns that have been hiding in complex multi-parametric data, and it can help. Bring out what we don't see. And moreover, it can help us understand a little more disease processes by looking at these subtleties of structure, function, and pathology, such as amyloid deposition in Alzheimer's disease. And they really complement human readings. And a lot of times our radiologists are worried that the machines are going to replace humans and so forth. No, they don't. It's just that the humans have to be used to read these new indices right now. And instead of looking at the image and trying to figure out whether or not you have the mutation, or instead of looking at the image and trying to predict whether or not this patient is going to do well, you just read a bunch of these instruments that give you an indication of whether or not you are expressing a given imaging pattern, and you have to interpret things differently. We have more data right now, more biomarkers, and I think machine learning plays an important role and will play an important role in reducing all this complex data down to a clinically feasible panel of maybe 5, 10, 20, or 30 biomarkers that have been informed by data. So you don't want to extract arbitrary values, but if there's a pattern of brain atrophy and functional change that we have seen in people with prodromal Alzheimer's disease that is predictive of progression, then you want to extract that pattern. So I think machine learning is going to help us to distill this complex data down to a small panel that hopefully 20 years from now you take your MRI, it's analyzed in the lab, and like you have in, say, blood clotting factors or your cholesterol test or everything else, your MRI test will be a set of 10, 20, 30 numbers that capture a lot of these characteristics that we want to know. I would like to thank a number of people in my lab. This might be a slightly obsolete picture. This is behind our lab, actually. We're an urban campus, but we have the privilege of a little pond there. So this creates the false impression that we're in the countryside, but it's cute. And feel free to visit our website and learn more about what we do. Thank you so much. I'll take any questions if you have.
Great talk. I thought the work on the infiltration signature and how that aligned with recurrence was very interesting. Could you tell me a little bit, how was this model developed? Were you actually looking at recurrence to come up with the signature?
Yeah, so we followed, this is a strict study, that's why it's only 90 patients. We have about 500 glioblastoma patients, but so we went back and we selected the baseline scans that had all of the modalities. Number two, we followed these people. These people are followed. We're lucky at Penn, and we have advanced MRI both at baseline and at follow-ups. So there are people who came back and they had recurrence. Some people never come back. Some people, unfortunately, pass away in the meantime. So these are people, the subset of patients who had recurrence. But sometimes there's what's called pseudo-progression. So the treatment effects that mimic recurrence, they're not recurrence. So you don't want to rely on the images. So we selected the people who underwent second surgery. And so we had the tissue, and there was pathology-proven recurrence. And we also had to exclude the people who did not have gross total resection. So a lot of times, for various reasons, some of the tumors left back because of its proximity to eloquent cortex and so forth. So these are not good cases for us to analyze because, of course, you have a perfect predictor of recurrence is where you left the tumor. So you want to. So we had 90 patients total that met all these criteria. So it's a very strict criteria, and then we used those initially in cross-validation. Initially it was 65 or so. We used 5-fold cross-validation, and then we used a prospective subsequent set of the remaining ones.
Great, thank you.
Yeah.
So I'm wondering if you could tell us some more about this study with 20,000 patients. Is that longitudinal?
how long will patients be followed? Is there any serial imaging or just one image?
Yeah, this is existing data. So we've been collaborating for 20 years or more with different studies, and now there's a lot of free data. We just incorporated some of the UK Biobank. So we put together all this data from existing studies. Some of them are studies of interventions for hypertension. Some of them normal observation studies. Some of them are longitudinal. There's a couple of longitudinal studies. Of course, ADNI has longitudinal data. The biggest longitudinal study we've been collaborating with is the Baltimore Longitudinal Study of Aging, which is something like 600 people or so who are followed up over many, many years. It started in '94. I was a postdoc. I was involved in that. So they've been following a lot of these people. It's an amazing longitudinal study with imaging, and it keeps adding imaging as modalities become available. Amyloid imaging has been added, tau imaging has been added. They have extensive cognitive exams and progression from normal to MCI and to AD. So some of the studies are longitudinal. Like the Cardia study is longitudinal. It's at younger ages. It's ages 45 to 65. So that is accumulating data. Not all of them are longitudinal, though. So one of the things we want to do is derive these patterns from this big sample cross-sectionally, and then go back and evaluate them in the longitudinal study to see what they mean. What does it mean to be in that part of the brain chart of the coordinate system? What does it mean in terms of your expected trajectory? And there's some interesting methodological issues there in terms of probabilistic models of trajectories and so forth.
Thank you very much. Very interesting topic.
Thank you.
So in a study like this, when you have 20,000 images studies, that's not really the question. But when you have a small population that you had of 100 or 150, how important is it that every scan is collected either on one machine or have the same MRI settings? So the variability from scan to scan in order to be able to be predictable.
Right. So the BLSA, the Baltimore Study of Aging, is a good example. It was initially started in 1993 or so, and it was very particular about keeping the same scanner, the same protocol. Of course, if you scan everybody in the same scanner with the same protocol and with no upgrades and things like that, you get consistent images. It soon became obvious that, number one, your results don't generalize to other scanners. So sure, whatever you learn from, if your goal is to understand some disease processes, is fine. But if you want to come up with a biomarker, it has to generalize across scanners and protocols to some extent. But also within the same study, the Baltimore study, as the scanners were upgraded, its imaging was done initially at Hopkins for several years. I was at Hopkins as well, and at some point I was talking to my chair, then Eliezer Hooney, and then he says, "Well, so what do you want us to do? To keep the same scanner forever? We can't. Clinically, we have to upgrade." So even the same study was not feasible to continue on the same scanner. So we have to come up with, at that point, I realized that we can't just keep doing that, and we have to come up with statistical or image analysis methods. And there's a lot of this stuff we've been working on, harmonization both at the image analysis level and at the statistical level. And that's how we put all the data together across scanners to be able to get something that works across multiple scanners and protocols. Clearly, the noise is higher, the variability is higher, but at least you get something that you know will reproduce to another scanner. But if you have a homogeneous protocol within the same scanner and the same protocol, of course it's better. You can detect effects much more effectively.
Alistair, I just want to say this is a fantastic study. I have two questions. The first of which, how would you account for newer techniques of doing surgery, like neurosurgeons doing LIT, etc.? And the second question is, do you think there is some role for something more quantitative instead of RCBV, etc.?
Into what?
Something that's more quantitative. So for instance, here at Case we do MR fingerprinting,
Yes.
which is more of a quantitative method of imaging. Do you think there's a role for using this system in fingerprinting?
Yeah, I mean, certainly there is a role, to start from the second one, there is a role, of course, if you have better features or different features, you'll do better. I mean, machine learning and analytics comes on top of what is available. So if you have a good way to obtain, like in fingerprinting, to obtain more rich imaging measures, that is likely to make this analytical estimations better, to leverage this information. So there's, and the same thing applies to the first question. If there are new surgical techniques, and I don't know what you refer to exactly.
Laser ablation.
Oh yes, yes, of course. The more of this you have, the better off you are, because these predictive tools, at some point, you say, okay, I can predict all these things, but what do I do next? If the approach is to just resect the enhancing part of the tumor, okay. But if now you start to use this more precise and essentially extensive but targeted treatments, you can start targeting the tissue that needs to be treated and sparing the tissue that should not be treated, or it's better to leave it alone. So I think the more of these new methods come up, the better off the more useful these tools are going to become.
Yeah, I was trying to formulate this as a question. It's more of a comment, I think. The things that you presented here, I see a huge potential opportunity for all kinds of other neurological disorders, like epilepsy and stroke, and even neuropsychiatry applications. Especially in the latter, the problem is that the effects of treatment and things are very delayed, and I just wonder how that affects. I mean, you have this as well in the cancer recurrence. I mean, the treatment takes a long time. How does that affect the effectiveness of the prediction when things are so far out into the future? The impact.
Well, I mean, if to some extent it makes it exciting, because if you can predict something of the future, it's great, right? So, but of course, it makes the studies longer and more difficult to perform. So in schizophrenia, we're collaborating with a number of groups now. This consortium has, we hope to grow the data set into 3,000 or so. Some folks in Europe particularly, but also in Brazil, they have first episode people who present at the clinic, and at some point these people progress. I mean, it takes years before you actually find the outcome, but there is data, and we want to look at that to see if at first presentation there are imaging patterns that can predict ultimate progression of these people to schizophrenia versus to regression back to normal or sort of normal. There's also a couple of studies we participate in. One of them is called the PNC, the Philadelphia Neurodevelopmental Cohort, which is led by Raquel Gore and her group in psychiatry, and they have collected about 1,500 scans. It's primarily cross-sectional, but these are adolescents, ages 8 through 21 or so, and they are following them. Some of them have been followed up. So we want to see now if we see these patterns that we learn from patients who develop neuropsychiatric disorders, if we see them in adolescence, which is where you would want to see them if you were to intervene. So I mean, it's still under these studies are under development, but I think having a long window of the effect is in some ways is a good thing, because you can do something. In glioblastoma, there's little you can do in practice right now.
I guess another aspect is that when you have a tumor, you see it and you have a quantitative way of assessing the outcome. Things like schizophrenia and other neuropsychiatry disorders like that are a little less specific, right? It's a questionnaire, it's a subjective assessment. So how does that impact? This comes up in our world pretty often. So how does that impact the sort of the specificity of your results?
Well, this heterogeneity is exactly what we want to dissect right now. And this is the big problem. People come to the clinic, and schizophrenia is a great example. I didn't show these results because they're not published yet, but it's very, very exciting stuff. So people come to the clinic and they present with schizophrenia, or at least clinically they're categorized as having schizophrenia. If you look at their scans, their images, even structural scans, about 38% of those people, more than a third, they have brain anatomy that's very different. The 2/3 of them have a typical frontotemporal pattern that I showed here. And both us and people like Paul Thompson and the Enigma Consortium have shown these patterns. We never appreciated that about a third of the people do not have or contribute to these patterns. And I showed these results knowing that 1/3 of the people are not there. And now we know that these people have something different. And so I think that this heterogeneity is actually what makes these methods even more exciting, because you can tease out the people who have a different pattern, and potentially they might benefit from a different treatment, or they might have a different outcome, or a different disease altogether. That should not be called schizophrenia, but should be called something else. So I think it's an exciting time. We'll learn a lot now from these methods, yeah.
So my question is more on the brain tumor work which you presented. Very exciting. So my question is, you mentioned about the clinical trial that you guys started on the recurrence project. Where else do you think, in terms of clinical trials in the near future, where we could apply some of these techniques and sort of, take it to the bedside, where radiologists and surgeons can start to use some of these methods in clinical practice?
Your question is where
else? So, recurrence, I think that's a great example, because, they can go in and, like you said, they're going in and radiating more, which they didn't used to do before. Where else do you think, given that brain tumors have such limited options in terms of treatment, where else do you think we could make a difference with some of these techniques?
Yeah, so it depends on the disease, of course, because the treatments depend on the disease. But in glioblastoma, there's a number of different treatments that are being tried right now. And there's some immunotherapy trials, there's all sorts of things. Again, there are two issues here. The common saying is that none of these treatments works or has worked. Well, sure, but how did we evaluate them? If you evaluate them in a small number of people who are heterogeneous, your chances of finding something that works are slim, because it has to be a pretty dramatic effect. Maybe it worked for some of the people. So I think the biggest bet would be if you try to dissect this heterogeneity, and that's where I think imaging has a role, and understand people who have this type of disease versus that type of or subtype of disease might be more likely to respond to treatment. I think that's where the biggest gains are going to be in terms of both clinical trials and also precision diagnostics. For example, people who are predicted to be very poor responders, there's no reason to wait for a few months for them to fail. Where the tumor has become more heterogeneous, it has progressed, the patients have undergone radiation therapy and chemotherapy, they've used most of the therapy they can take, and they become more vulnerable. It's better to put them in an immunotherapy trial up front that might work better for them. So I think all these opportunities start to appear right now, and we don't know exactly how they will influence the decisions. The other area where I think we're going to see a lot, and imaging plays a very important role, is characterizing the spatial heterogeneity of the tumors. Heterogeneity is very big, and right now they take one tissue sample and they call the tumor positive or negative. This is absurd, because we know that most tumors are both positive and negative. They have mutations in certain parts and they don't have the mutation in other parts. So what does it mean to treat a tumor with a monolithic treatment if you know that's a mix of things? So again, imaging can, now clinically it's nearly impossible right now financially to support multiple analysis of tissues, maybe at some point, but imaging I think can highlight the heterogeneity very nicely. We have to perform studies to show that the heterogeneity in imaging that we see in imaging maps really to molecular heterogeneity, and that takes some time and some effort, but I think it has to, because certainly, as I'm sure you've seen, you know very well, in glioblastomas, you have this superior part of the tumor that might look completely different from the inferior part of the tumor, and they most likely have different mixes of mutations. Okay, thank you so much.